The first part of Chapter 19, which is on genetic technologies, is going to focus on the difference between recombinant DNA and transgenic organisms, and really understanding how those are related, and then some applications of recombinant DNA technology, and then what are the methods involved in constructing a recombinant DNA molecule. So this is a starter that we did in class about pig manure and the EnviroPig. So the term biotechnology refers to the use or alteration of cells or biological molecules for specific applications. And a transgenic organism is one that has DNA from different species. And so we often use transgenic organisms in biotechnology. Recombinant DNA means that it's DNA that comes from more than one type of organism. And so basically, a transgenic organism has recombinant DNA inside it. The reason we can do this is because of that quality of the genetic code that is universal, okay? That means all organisms use the same genetic code. And so because of that, we are able to do some of this recombinant DNA technology. Now, one of the things that arose because of taking genes from one organism and inserting it into another to make various products, maybe such as insulin or other types of useful drugs, is a lot of that now can be subject to being patented. And so one of the things that they have to look at in order to have a patent protection, the transgenic organism must be new, useful, and non-obvious, meaning it has to be something that nobody's come up with before, that is used for a very specific purpose, that's useful in some way to help humans. And so a lot of law going on here, patent law, that has to be subjected to any kind of, when you look at a lot of different medical research, genetic research, whatever, they have to look at that. It has to be useful as a tool for research or novel or improved drug, such as a diagnostic test or drug. This just gives you a little bit of history on how patenting, especially in relation to genes and genetic sequences, has progressed throughout the years, and all the way up it goes as far as 2013. And you can see in 2013, the Supreme Court declares that genes are unpatentable, which that allows all researchers then to be able to share and utilize similar sequences to do their research. So recombinant DNA technology then adds genes from one type of organism to the genome of another. This was initially done in bacteria to produce peptides and proteins useful as drugs. One of the main ones that we still use today is insulin, because we used to have to either get it from pig pancreases or cadavers or things like that. So it's nice now that we can have bacteria mass produce human insulin, because we've inserted the human gene for insulin into the bacterium. Sometimes recombinant DNA technology is known as gene cloning, so you might hear that as well. So how do we actually go about creating a recombinant DNA molecule? Well, first, we need what are called restriction enzymes. These cut both the donor and the recipient DNA at the same sequence. And these enzymes cut DNA at sites that are palindromic. That means same forward and backward. Think of the word like race car, spelled the same forward and backward. Well, these restriction enzymes find sequences that are the same forward and backward on both sides of the DNA strands. When they cut the actual DNA, it leaves these extensions called sticky ends, and you're going to see that in the next slide here. So you can see here, see how this goes G-A-A-T-T-C. Well, coming the other direction, it's G-A-A-T-T-C. So same forwards and backwards, that's a palindrome, and that would be a site that a restriction enzyme could cut. But it doesn't just cut it like straight down the middle like that. It cuts it in these jagged cuts, which then leaves these sticky ends. And now you can cut your donor DNA at the same location, and now it will fit just perfectly like a little puzzle piece right here. And you can just insert that DNA, the donor DNA, right into the host DNA. So besides the restriction enzymes, we also need what's called a cloning vector. A cloning vector carries the DNA of cells from one species into the cells of another. Now, a lot of times plasmids are used. This is what we used in lab, in our transformation lab, but it's a small circular piece of DNA. And it's found in bacteria, and usually it doesn't have genes on it that the bacteria necessarily needs to survive, but it oftentimes has antibiotic resistance genes on it, which are very helpful to the bacterium. Bacteriophages, these are viruses that infect bacteria. And then disabled retroviruses. HIV is a retrovirus, okay? You obviously wouldn't want to inject that into somebody, but any kind of retrovirus that they do, these are RNA viruses, meaning that they have to go backwards to use reverse transcriptase to kind of reverse transcription to make their DNA. So a lot of times they'll use disabled ones. They don't carry any infection with them, but they can be helpful in bringing that DNA in there. So we have to first of all cut the DNA from the donor and the plasmid vector with the same restriction enzyme, and then we mix to generate the recombinant DNA molecule. Then the modified plasmid gets introduced into a bacterium, and there it can be mass produced as the bacterium divides. And so you can see here we have our DNA isolated from the donor cell, and then the restriction enzyme is going to cut that DNA from the donor cell. Same thing, we have a plasmid DNA, and then the same restriction enzyme cuts it. Then you mix it together so they join, and then they get inserted into the bacterium here, and then anytime the bacteria divides, it is reproducing and making that, and so you can mass produce things such as insulin, and then of course needs to be purified and produced, but and then it's ready for use by humans. Now there's 3 types of recipient cells that can result from an attempt to introduce that DNA molecule into it. You might get cells that don't take up the plasmid, okay? Or you get cells that have the plasmid, but they don't contain the foreign gene. Like the foreign gene never got inserted into the plasmid. But here's what we hope you get, is that you get cells that contain plasmids with the foreign genes. All right? And usually one of the things they'll do to test that, remember those plasmids have antibiotic resistance genes in them, they'll try to grow the bacterium on a plate that has the antibiotic in it. And if your bacteria can grow on that plate, then that means it has the antibiotic resistance gene, which means it has the plasmid. So then you're hoping that it also has the gene that you inserted into the plasmid. So that's kind of one way they can kind of check to see if you at least got the plasmid in. Again, why do we use recombinant DNA technology? What are some applications? Study biochemical properties or genetic pathways of a protein, mass-produce proteins such as insulin. Sometimes it's just simpler and easier to use conventional methods, but if we can mass produce it cheaper, more cost effectively, then that's a good way to go. If you think of the various any of you that wear blue jeans, the indigo dye they have actually genetically engineered E. coli to produce that indigo that goes in there. So that's kind of always interesting. And here's a bunch of other uses of genetic recombinant technology, some of the various drugs that have been produced over time.